Did you know that we share 60% of our DNA with strawberries? Now, that doesn't mean that we're going to start sprouting leafy heads and turn red, but it does mean that at one point, many, many years ago, we descended from a common ancestor. So how do we determine the commonalities between us and strawberries? Well, this is where PCR comes in. PCR stands for polymerase chain reaction. It uses a special type of the enzyme DNA polymerase called Taq polymerase in order to create several copies of a target DNA segment. PCR has three main steps: denaturation, annealing, and extension. First is denaturation. In denaturation, the temperature is raised to around 94 to 96 degrees Celsius so that the double-stranded DNA molecule can split into two strands. In annealing, the temperature is cooled to around 60 to 65 degrees Celsius so that primers, which are small segments of DNA, can bind to opposite ends of the target sequence using complementary base pairing. Taq polymerase can only add new bases onto an already existing strand, so the primers provide it with a starting point. Finally, in extension, the temperature is raised once again to around 70 to 75 degrees Celsius, and Taq polymerase creates the new strand by adding complementary bases in the 5' to 3' direction. These three steps are repeated multiple times in a machine called a thermocycler until the desired number of copies are produced. So, how many copies can PCR actually produce? We can model the number of double-stranded target sequence molecules with the equation 2 to the n minus 2n, where n is equal to the number of cycles. 2 to the n represents the number of total double-stranded molecules that are produced, and 2n represents the number of molecules that have excess DNA on either side of the primer. After one cycle, we would produce 2 to the n double-stranded molecules, which is 2, and 2n, or 2 molecules with excess DNA. This means that we wouldn't actually produce any target sequence molecules. The same goes for the second cycle, but at our third cycle, we'd produce 2 target sequence molecules, and at our fourth, we'd produce 8. By our 10th cycle, we would produce 1,004 target sequence molecules. So why PCR? The copied DNA from PCR can be used in various ways. It can be sent for sequencing, put through a gel electrophoresis, or inserted into a plasmid, all of which will help us determine genetic commonalities. PCR, along with various other methods, have allowed us to determine genetic similarities between us and all kinds of organisms. So the next time you pick up a strawberry, think about how similar the two of you really are.